Now, in this lecture, we'll talk about the mechanism of action, how these antibiotics act on, and how these targets are used for antibiotics to mechanize to act on these targets to kill the bacteria. Now, if you look at the mechanism of action, uh, in the mechanism of action, they have a different mechanism of action as we already discussed in earlier lectures. Here we can talk about in detail. For example, some of the antibiotics, they act on cell wall synthesis like penicillin, cephalosporin, bacitracin, vancomycin. So this is the bacterial cell, this is the membrane, and when the membrane is targeted, then it will lyse the bacteria. So membrane is, for example, targeted like this. So it will lyse the bacteria. When the bacteria lyses, the cell will die. Now, in some cases, it inhibits the protein synthesis, so it block the translation process, right? And this translation process, when it blocked, protein synthesis will not form, and when the protein synthesis will not form, cell will again dead. And then we have inhibition of nucleic acid replication. In case of nucleic acid replication, several enzymes like gyrase, topoisomerase. So this is how the DNA replication will be inhibited, and so no DNA will be formed. And when the no DNA will be formed during the cell division, the no DNA is present, and the cell will stop the growth of the proliferations or the further growth. So these are some mechanism involved in antibiotics. Similarly, we have injury to the plasma membrane like polymyxin B, colistines, and bacitracin, then inhibition of essential metabolite like synthesis sulfonamide and trimethoprim. So they act on enzyme, some modification for some enzymes, so the enzyme may not act on, and so the metabolic processes will be blocked. So the key concept of this is antimicrobial drug resistance. Basically, drug targets rather certain essential functions of a microbes, and these essential function of a microbe is being targeted by antibiotics to kill the bacteria. Now, bacteria may also have certain genes which are not essential for the growth. Now, if you act on these targets, it's useless to make such molecule which can act on the target which are not essential or non-essential target. Therefore, all the targets should be such that they should have essential activity in the bacteria, so that it can hit the bacteria to inhibit through these molecules while you are designing new antibiotics, or while you are working on some molecule which we call them as the antimicrobial agent. Antimicrobial drug must not interfere with the essential function of the microbe's host. So this is very, very important. So while you're designing the molecule, you should be careful about there's no human analog in the bacteria, or the gene should not have any analog in the human system where this molecule may also target. So antibiotics should exclusively for the bacterial system, exclusively for targeting to a specific bacteria, should not target on the human cell. Now, sometimes people say when antibiotics are used, they should have a side effect. These antibiotics may have a side effect not because of it target to the human cell, or it target to the mammalian or the human cell. No, it target to the bacteria, good bacteria in the gut, and when the good bacteria are targeted, then they kill the bacteria, good bacteria also, along with the bad bacteria, right? So when the good bacteria are killed, it will affect the health of the human. So on the one side, you are inhibiting the infection, secondary infection, or any kind of infection caused by bacteria. On the other side, you are also developing like low immunity, you are also developing some other disease conditions because killing the good bacteria which protected human for such kind of disorders. Now, inhibiting the cell wall synthesis is in more detail. Cell wall bacteria is made up of the peptidoglycan. Now, peptidoglycan is made up of certain molecule which are linked through certain bond, and like N-acetyl muramic acid, N-acetyl muramic acid, and N-acetyl NEC and NAB. We call these two molecules, and these molecules are linked. The linkage is blocked, the linkage is broken, the bond is broken, right? So antibiotics such as the penicillin prevent the synthesis of intact peptidoglycan. right? Cell wall is weakened because of the breaking in the bonds. Uh, the target is actively growing cell. So the actively growing cells is stopped, right? And human cell lack peptidoglycan, therefore it will not act on the human system. So this is one of the advantage of these antibiotics. Uh, Targeting the plasma membrane, right? What is the plasma membrane? The plasma membrane is the membrane which is next to the peptidoglycan inside, right? And this the leakage of the plasma membrane also cause the lysis of the cell. So when they target to the plasma membrane, the targeted by the polypeptide antibiotics to the plasma membrane, then permeability will be increased, or permeability will be imbalanced or altered, and because of this endosmosis process, it can lyse the cell, and the cell will die. The loss of metabolites from the cells also, which can also cause inactive cells. Cells become inefficient to grow. So, right, all these things will taking place, and because of this, the cell will inhibited, our cell will killed or destroyed. Now, in case of inhibiting the protein synthesis is common between the prokaryotes and eukaryote, right? So, while the antibiotics is against the protein synthesis, it might also against the protein synthesis of the human, but this is not the case because nature has provided the different protein synthesis machinery in bacteria and in human. So, in case of bacteria, the machinery or the industry for the protein synthesis is 70s ribosome subunit, where you have uh, 30s and 50s ribosomes are present separately, which can bind the mRNA for upholding the protein synthesis process by bringing the tRNA, amino acid tRNA, a specific amino acid tRNA for amino acid to act on, and codon and anticodon get linked to bring the to get the amino acids sit on the specific codon on the mRNA, which is bind between the 30s and 70s ribosome, 50s ribosome. But in case of human, it is our mammalian system or eukaryotic system, it is 80s ribosome. So therefore, since it has a binding capacity only specific to the 70s ribosome, that is either 30s ribosome or 50. So some of the antibiotics they act on 30s ribosome, some of the antibiotics they act on the 50s ribosome. So therefore, human cells are free. I mean, they may not be attacked or targeted by these such antibiotics. Still, mitochondria have a 70s ribosomes, which is like a, in eukaryotes. Hence, there can be a damage to the host cells. So the only possibility of damaging host cells is that and since the mitochondria is the powerhouse of the cell, so it produces a lot of enzymes which involve in the energy productions, right? And if the ribosomes, if the mitochondria is lost or mitochondria is affected, then it will definitely affect the host health and host cell. For example, chlorphenicol, 50s subunit, erythromycin, streptomycin, tetracycline as 30s subunit, streptomycin, erythromycin, they're all as antibiotics. They act on 70s ribosome complex, either 30s or 50s ribosome, either through blocking the protein synthesis, through blocking the tRNA, or through upholding the mRNA, or translocation, blocking the translocation, or blocking the peptide bond formation. So all these mechanisms are involved in inhibiting the protein synthesis. Now, if you look at this figure, in this picture, you will find that I can explain the three-dimensional detail of the protein synthesis side, showing the 30s and 50s. So this is the 50s ribosome, this is 30s ribosome, and this is how the protein synthesis takes place. So this is 5' to 3' mRNA. Now, in case of prokaryotes, as I told you earlier, that protein synthesis and mRNA synthesis, that is transcription and translation are the coupled process. So this is what I mean, the coupled process, which is not in case of eukaryotes. So this is how the mRNA synthesis takes place, as well as the protein synthesis takes place. Now, the growing peptide, so this is called tunnel, where the in this tunnel the peptide bond formation takes place, and translocation takes place, and it all goes through these proteins through this tunnel, and they form growing peptides. Now, so this is the first step. Now then we have this is how growing peptide takes place. So for example, this is the streptomycin, and this streptomycin change shape the 30th ribosome. So when the 30th ribosome shape is changed, like compared to this and this, right, then the mRNA is slightly kinked, and because of this, the binding of the tRNA here is not possible, is not taking place. And in case of translation process, streptomycin intervene, and they manage to block the translation process through growing peptide. So now this is how the chromphenicol bind to the 50th ribosome here, and it inhibits the formation of the peptide bond. Now this is peptide bond formations when the chain come to the new tRNA, bring the amino acids. So there's a binding between these two to form peptide bond to form further chain, further growing chain. Now, so chromphenicol bind to 50th subunit, and then it block the bonding between these two amino acids, that is the amide and carboxy link. So there's a no amide bond, there's a no peptide bond formation takes place. So there's a no linkage, and then there's a no linkage, no growth. So there's a no growth take place. Otherwise, 1, 2, 3, 4, 5, 6, it should be like 1 like this. So it should have now 7. Similarly, it will translocate. Now, this is how the tetracycline act on 30th subunit again, and it interfere with the attachment of a tRNA to mRNA. So this will help the cell. I mean, rather this will discourage the cells to bind with the mRNA to tRNA binding, and mRNA and tRNA binding is blocked with the tetracycline because it binds to the 30th subunit. So this picture is basically clear cut, showing that how these protein synthesis will be blocked on the ribosome. Then we have inhibiting the synthesis of the nucleic acid. Now, nucleic acid is again very important, because in a cell, if you look, this is the cell, and this is the bacteria nucleic acid, and this nucleic acid is basically involved in the replication process. So you can have a second copy. Before it forms a second copy, it transfer into like this. So from a single bacteria, you can form 2 cell and so on. So this cycle is on. So this is called the growth of the bacteria. Now, what is important for antibiotics to stop the growth, right, or to kill the bacteria? So both the way it works. So some of the events like protein synthesis, when it is stopped, it will stop the growth as well as it will kill the bacteria. In certain cases, it will not kill the bacteria, but it will stop the growth. We call it bacteriostatic, right? So inhibiting the synthesis of the nucleic acid by interfering with the DNA replication and transcription process, because when the DNA is because nucleic acid, both like mRNA is nucleic acid as well as DNA is a nucleic acid. So both replication and transcription both are blocked, or both are affected by these antibiotics. Now, so by interfering with the DNA replication process, several enzymes of the DNA replication process are involved in that, which are which are basically inactivated during the replication process in the presence of that antibiotic, right? Like topoisomerase, gyrases. So they will not. The function of these topoisomerase and gyrases is this. For example, this is the DNA. It is. So they start making unwinding the DNA in this form of the DNA. It will not replicate, or it will not transcribe. So to get the replication process or later on transcription process, what is important is the DNA should be like this. So this is called uncoiling, right, or unwinding of DNA. Now the unwinding of DNA or uncoiling of DNA is important for the replication process, because what you need for replication process is this this. So when the replication takes place on the enzyme like this, it will synthesize another strand here like this. Right. So this is how the replication process starts. Now, so what this antibiotics basically do, or this specific antibiotic does, it block the replication process by enzyme called digase or helicase. In certain cases, helicases, digases are topoisomerases. What is the function of topoisomerases? It basically insert the negative supercoil. It like uncoil it, negative supercoil. So by putting the negative supercoil, it open like this. This is all closed, see here, but this is become open. So this opening is because of the negative supercoil. So the enzyme which make the DNA to open, to uncoil, to unwind, right, or insert the negative supercoil is being targeted by these antibiotics, so that the antibiotics, the DNA will not form of this kind of a structure, rather DNAs remain in coiled form like this. So this DNA, no replication, no transcription, no replication and no transcription, right, and this is what is done by the antibiotics. So bacterial topoisomerases in DNA and RNA polymerases are the block. Because of this reason, the replication process will not take place. Now, it inhibiting the synthesis of the essential metabolites, right. So what are the essential metabolites? Essential metabolites, as I have told you earlier in one of my lectures in the same series, that enzymatic activity produced by certain metabolites which are essential for the growth of, we call them primary metabolites, primary product, byproducts, right, which are essential for like the protein, like lipids, like carbohydrates, fats, and so on. So they inhibit the essential metabolites, and by inhibiting the essential metabolites, they do, they kill the cell. The cells will not able to grow, they will not able to proliferate, they will not able to further process their growth. So enzymatic activity competitively inhibited by these antimicrobial agents, by an anti-metabolites, exhibiting close resemblance to the actual substrate. So they kind of make molecules which can have a close to the substrate, right. Just for example, I take an example of the antiviral drugs. So most of the antiviral drugs, they are nothing but they are the base analog. Base analog means like they have a nucleotide base analog which they will given to the patient, given to the site of the viral infection. What it does, it compete with the incorporation of the bases, nucleotide bases, so that it will, when they incorporate because of competitive inhibition, then the next base will not continue, correct, and then it will block the DNA synthesis. Then, so this is similarly the antimetabolites which are analog of the metabolite, essential metabolites will compete, and it will not involve or acted upon by the enzymes responsible for the conversion of these metabolites for the further essential product, and hence it will kill the cells, it will destroy the cells. For example, antimetabolites, sulfonamide, this is called the sulfur drug, very common, right, Septron, trimethoprim, right, and they are the para-aminobenzoic acid. Para-aminobenzoic acid is a base analog which can compete with this, and the DNA replication enzyme will not recognize it, and it will block the synthesis of the nucleic acid. Beta-lactam antibiotics are basically very common antibiotics, and maximum maximum antibiotics which are present under the class of beta-lactam. And beta-lactam antibiotics are basically of different classes, from generation 1 to 4, cefepime, and then we have certain beta-lactam inhibitors combined in association with these beta-lactam antibiotics, we call the 4th generation of antibiotics. Or then we have carbapenems, meropenems, right, then these are all beta-lactam antibiotics. Then we have several other like colistines, which is not beta-lactam antibiotic, which is polymyxin B, but these antibiotics are all acting upon the membrane. So the large group of these antibiotics, they have a ring-like structures, and these ring-like structures, this ring-like structures is blocked, is broken, and after breaking this link, they will inactivate the function of antibiotics, whatever antibiotics are present here, right? And this ring is blocked, right? After blocking the ring, the activity is lost. So this is how the antibiotic degraded. Now, degradation of antibiotic or beta-lactam ring of antibiotic is basically responsible for the inactivation of the antibiotics, and such kind of antibiotics will not act on the target. It will not bind to the penicillin binding protein. So PB PBP is basically will bind only the intact antibiotics, intact antibiotic group. So this is how the beta-lactam antibiotics work. So the primary target is a bacterial cell wall, and they bind to the penicillin binding proteins and block the cross-linking of peptidoglycan, leading to the cell lysis. So this is the mechanism of the beta-lactam antibiotic. So several enzymes which are also produced from these beta-lactam antibiotics, we'll talk later, which enzymes also degrade this beta-lactam ring and so become resistance against these antibiotics to the bacteria. Now, the classes of the beta-lactam antibiotics, we have different classes like penicillins, are example is penicillin G, penicillin V, ampicillin, amoxicillin, oxacillin, and piperacillin. So like many more examples are also there. Now, what are the key features and what are the spectrum of this antibiotic? So first discovered beta-lactam antibiotics, that is penicillin from Penicillium notatum. It active against the gram-negative bacteria, gram-positive bacteria, extended spectrum form cover some of the gram-negative bacteria as well. Since peptidoglycan is more thick in gram-positive bacteria, so it is more of like acting against the gram-positive bacteria, but also act on the gram-negative because thin peptidoglycan is also present in gram-negative bacteria. Then cephalosporins is of a different generation, like we have first generation cephalosporin like a cephalexin, then you have a cefoxim, second generation, then we have a third generation like a ceftriaxone, then we have a fourth generation like cefapime, and then fifth generation is ceftriaxone. Now, this generation is basically the advanced form of the antibiotics with slight modifications, changes as and when required when the resistance start developing. For example, when the resistance start developing in first generation, so it comes next to the second generation. So, and this is how the different line of antibiotics given to the patients by physicians as given the first generation, if it's not effective, then second generation, third generations, and so on. The classified, they're all classified by generation with each generation like gram-negative coverage, they generally improve, and while gram-positive coverage varies. So these antibiotics are good for both gram-positive and gram-negative, but mostly they are gram-negative. It covered the broad spectrum gram-negative, and every second generation is better than the previous generation. Carbapenems are again the new drugs after the fourth and fifth generations. They come, and we call them as a last resort of antibiotics because of the fact that they can act on even the metallobeta-lactamase producing enzyme. Metallobeta-lactamase producers, that is enzymes which degrade the class D and carbapenemases, are metallobeta-lactams also. Besides serine beta-lactamases. So there are two types of enzyme, one we call the serine beta-lactamase and another we call the metallobeta-lactamase. Both have slightly different mechanism of action in terms of like cleaving the antibiotic or hydrolysis of the beta-lactam ring of different group of antibiotics. But the carbapenems are basically more advanced form of the enzymes carbapenemases, which can cleave even the carbapenems, which is the last resort of antibiotics in the beta-lactam category. Like imipenem, meropenem, which are highly used, frequently used in several setting of the hospitals, but OPD, IPD, ICU, and ICU, and they are broad spectrum gram-positive and gram-negative anaerobes often reserved for the resistant infections. So they are given only when the high resistance is developed in the cell, and then it is given. Then monobactam aztreonam, the narrow spectrum, mainly effective against the aerobic gram-negative bacteria, useful in patients with the penicillin allergy. So this is another class of, again, this is the broadest spectrum, considered to be broadest spectrum antibiotics, but it is a narrow spectrum because it mainly affect on the against the aerobic gram-negative bacteria, useful in a patient with a penicillin allergy. But earlier when it was used for the endium producing strain, sometime it might also work against the endium producing strain. So that may be considered also in various gram-negative anaerobes having the class A and class D producing bacteria. Beta-lactamase inhibitor used in combination, which is the most recent development where the several inhibitors. Now, what does these inhibitor means is, so this is, for example, beta-lactamase inhibitor. Now, this beta-lactamase inhibitor is basically inhibiting the ring amide bond, this ring and this cleave, and so this ring is become open. Now, this inhibitor, beta-lactamase inhibitor, which basically help in like so this is enzyme beta-lactamase enzyme. It may be serine beta-lactamase, it may be metallo-beta-lactamase, right? In both the cases, it will cleave it. Now, this hydrolysis which is taking place because of this enzyme. So, what does the BLI does? BLI block this, and by blocking this, it protect this hydrolysis, right? And it protect this hydrolysis, and it will remain intact. Now, when the antibiotics remain intact with the beta-lactam ring, it will act on bacteria and kill the bacteria like this. So, this is the simple mechanism of inhibition bacteria and protecting the uh, protecting from the resistance against these antibiotics. Now, when the resistance develop, these beta-lactamase inhibitors, right, there are n number of inhibitors are in market like sulbactam. So, these are the clavinic acid is one. So, what is the clavinic acid, sulbactam, tozobactam, or avibactam do? They block the beta-lactamase enzyme, and by blocking the beta-lactamase enzyme, it inhibit the action of beta-lactamase enzyme onto the antibiotic beta-lactam ring. So, when it protect the beta-lactam ring through inhibiting the beta-lactamase enzyme, it make the make the antibiotic more efficient to kill the resistant bacteria. So, they are the non-active alone, right? And this is how it protect the beta-lactamase enzyme. Now, similarly, we have some more. So, this is the Ley and Ambler classification of beta-lactamases. So, these are the two classification of beta-lactamases which are commonly used for identifying classes in terms of like A, B, C, group 1, 2, 3, and so on. So, Ambler classification, the molecular based on amino acid sequences. Class serine beta-lactamases. Then we have class B metallo-beta-lactamases with the zinc divalent ion, and then class C MC type serine enzymes, and class D type serine oxygenase or oxenzyme. We have the oxyserenase is present. Now, these are the four category of enzymes by the Ambler classification. This was done by, I think, Jacob, and this is done by the Bush and Jacob. And this is Ley's classification is basically functional based on the substrate profile and the inhibitor responses. Like group 2, is a penicillinases, ESBL, some carbapenemases like TEM, SHV, CTXM, KPC. So, this is the example of both, but they come under the group 2 in case of Ley, and in case of beta-lactamase class B, in case of Ambler, it is a NDM, WIM, IMP, which are metallo-beta-lactamases. We call group 3 in case of Lay's classification, beta-lactamases. Class C MC type serine beta-lactamases comes under group A in cephalosporinases and MC. And example is like MC, beta-lactamase, CMY, FOX, and DHA. This is an example which, so Lay's classified them as a group 1, and Jacob and Bush classified them as under the Ambler classification, classified as the class C. Similarly, class D is group 2, subgroup of group 2 and group 2 and group 4. We call miscellaneous unclassified rare enzymes, like some uncommon beta-lactamases, which are uncommon, not characterized, and they are not very common beta-lactamases. But we like OXA type of classification sometime. Then we have OXA derivatives of classification, of the OXA derivative class, which are less active, or which are we call them as a low expression enzymes. So having OXA alone may not have much effective developed resistance, and that can be inhibited by several imipenem, meropenems sometime. But having the OXA with NDM or some other metallo-beta-lactamases like IMP or WIM, it is more severely severe resistance showing against that. Now, antimicrobial resistance again. So in a nutshell, the ability of the microbes to resist the action of antimicrobial agents called antimicrobial resistance. The major problem of the antimicrobial resistance, you should also know about it caused 4.95 million deaths in 2019. And it can cause up to 8 to 10 million deaths by 2050. So the mechanism of resistance, whether it is through beta-lactamase producing, or through aminoglycosides, or through enzyme modifications, or through modification in the nucleic acid inhibitor enzyme inhibitors, it may have like different kind of a mechanism, maybe efflux pump blockage, or maybe have sort of OMPs broken, OMPs degradation, or OMPs fragmentation. Make all these mechanisms cause the resistance in the bacteria against the antibiotics. So having that is PVP binding proteins, maybe penicillin binding proteins may be blocked. So having all these resistance develop in the bacteria, wide varieties of bacteria, and they are spreading. Now, why this data is showing here as a 10 million people die by 2050, it is because of the spread of these resistance. Now, you can ask a question, how resistance is developed, or how these, spread takes place with these. So these are all genes that we talk about, like TEM, SHV, CTXM, KPC, NDM, WIM, and there are number of variants they have. All these genes are called ARGs, antibiotic resistance genes. All right, and these ARGs are basically the gene responsible for development or for developing the resistance in the bacteria. Now the question is, the spread is because see, this population is going to die because of the fact that large number of bacteria, they are harboring, they are acquiring the resistance, because the resistance is sort of immune system or defense system of a bacteria. They protect themselves from. Now, when they acquire these resistance through these gene in the form of plasmid, so if they are having if the genes are present on the plasmid, right, or chromosome, so if they are present on the plasmid, then the mechanism of spread is called horizontal gene transfer. We call through mobile genetic element. We call mobile genetic element, MGE. Now, if it is on the chromosome, then we call vertical gene transfer, where when the cell divide, it will divide, right? So cell will divide and they will remain in its own. For example, if E. coli is dividing, they form E. coli, or if Pseudomonas aeruginosa is dividing, it will remain in Pseudomonas aeruginosa. So in this case, you have a chance of spreading. In case of plasmid, you have a chance of spreading through mobile elements, through jumping genes, and the process is called horizontal gene transfer. So the most dangerous part of this is the horizontal gene transfer, or the acquiring, or the having these resistance marker, or ARGs on the plasmids, so that the plasmids can move from one cell to another cell. So I think this is all about the antibiotics and antibiotic resistance. and resistance mechanism and their mode of action and how the resistance develop, what is the risk factor of the resistance and how we can control. Now, it's not a part of the topic how to control the resistance, but I think let's in the 1 minute I can share you the best thing is that to control the resistance mechanism is the minimum use of antibiotics, because maximum use of antibiotic put a pressure on these selection pressure, and that led the bacteria to acquire these infections. So I think we should be careful about it. And with this, I thank you very much for this listening my talk. And these are the some references, reviews, books where you can go through to understand more about the antibiotic resistance, about antibiotics, and some more papers are also available. Okay, thank you very much.